Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. A couple of weeks ago, I had the privilege to attend a meeting in Hamburg, Germany. It's called the Blankenese Conference, and it was something that was sponsored by the Jung Stiftung, or the Jung Foundation there in Germany, which is an advocate for biomedical research. And at the meeting, I ran into Samir Andalusi, and he presented some data from the great compendium of work that his lab has put out, including two recent papers. And we are fortunate today to have Xiuming Liang, who is a co-first author, a co-corresponding author, and co-last author, depending on which paper you're looking at, of these two papers that recently were published in the Journal of Controlled Release and in Nature Communications. So Xiuming is a research specialist at Karolinska, the Karolinska Institute in Sweden. And as some of you might know, the Institute, Samir's lab has a collaboration with Evox. And so Evox is an EV-based company that is doing some very innovative work as well. So Xiuming, thank you so much for joining us today and for being willing to present this work. I don't know how you're going to fit it into an hour, to be honest. There's so much here, but it's very exciting, and I'd like to hand the screen sharing over to you.
Thank you, Ken. Thank you for the invitation. It's a great honor to be here to talk about the work in our lab. So I will share my screen. So can you see my slides now?
This looks great.
Yeah. So today's topic is about the intracellular delivery. So in our lab, we are focusing on delivery, like functional therapeutics or genome editors. So in terms of intracellular delivery, we all know that like the cell membrane constitutes the natural barrier for large proteins or molecules to get inside of the cells. So for this reason, people have developed some vectors, we call non-viral vectors, so such as lipid nanoparticles, polymers, or other lipid-based delivery vectors. So these particles will encapsulate the large proteins and will be uptake by the recipient cells and by endocytosis. And then the carriers will release the functional cargoes into the cytosol of the recipient cells to mediate a lot of activities of the recipient cells. So there are a second type of the vectors, they're called viral vectors. Three of them are very well known. One is adeno-associated virus, and the second one is the lentivirus, and the last one is the adenovirus. So you know the AAVs, they are single-stranded DNA virus, and they have a limited genome package capacity. So its genome is around 4.7 kb. And for adenovirus, it's more compatible because the transgene can be up to 35 kb. And the last type is lentivirus, and it can have the transgene and other components up to 9 kb, but this transgene will be integrated into the human genome after transduction. So it will support the long-term gene expression. So even though the vectors developed are very good for some cell types, but they all have their own pitfalls. For example, for LMPs, they have poor endosome escape in the recipient cells. They have limited tissue specificity. Also, they have very short half-life in the circulation. For adeno-associated virus, like they have limited cargo capacity, as I mentioned before. So the genome is around 4.7 kilobase. So also some people already got infected by the virus, so they have generated pre-existing immunity. So this will have an issue for the administration of the AVs. Also, these AVs will have the persistent expression in the recipient cells. So this will cause the side effects of targets. For example, if you deliver a CRISPR-Cas9. And apart from the synthetic nanocarriers, also the viral vectors, can the EVs, they are naturally derived from the body cells, be good carriers? So if they can, they have a lot of benefits, like they are natural, they have low immunogenicity, and they are friendly to the body themselves. So the EVs mainly contain three parts: exosomes, apoptotic bodies, and the microvesicles. So here we take the exosomes as example. So it is formed like this in the late endosome. Some materials in the cytoplasma will be packaged, and the endosome will have the back fusion to form multiple small bubbles in the large vesicle. We call it multivascular body. So if this big bubble fills with the plasma membrane, the small bubbles will be released to the extracellular space. We call it exosomes. So the exosomes can carry functional elements such as lipid, protein, or nucleic acids. They can mediate the cell-cell interaction. So they can bind to the receptor on the recipient cell surface to trigger some cell signaling, or they can go endocytosis pathway to release the functional cargo to mediate a variety of cell functions. So if we would like to use EVs as promising nanocarriers, there are some challenges related to the biology or engineering. Here, we listed a couple of limitations. We cannot like solve all the limitations. In our study, we focused on two of them. I highlighted here. So one is the efficient cargo loading, because you know the naturally derived EVs normally they don't have a lot of like functional protein or the protein of interest. So if you want to deliver some functional protein like a CRISPR or Cre, you need to active load them into the EVs. So the second part is the poor endosome escape in recipient cells. You know that if the EVs go endocytosis pathway, majority of them will fuse with the lysosome and get degraded. So increase the endosome escape will be a key point to deliver functional cargoes intracellularly. So to solve these limitations, we have some strategies, and then we have the study in paper one. So in this study, we integrate EV sorting domain and also self-cleavage inting and the phylogenic protein in the delivery system. The aim is to enhance cargo loading and at the same time boost endosomal escape. So our strategy is like this. We use EV sorting domain, that's traditionally used CD63, to be fused with a cargo protein like Cre or CRISPR-Cas9. And here we have a novel strategy. We put the self-cleavage small element inting in between this EV sorting domain and the cargo protein. So this inting is a mutant one, and it will perform the C-terminal cleavage at 37 degree When the pH drops, it will boost the cleavage. So during EV biogenesis, so the cargo protein can be cleaved, as shown here. If we decorate the EV surface with the phylogenetic protein VSVG, the engineered EVs will fuse with the endosome membrane after endocytosis in recipient cells. And this is mediated by VSPG. So then the functional cargo like Cre or CRISPR-Cas9 will be released into the cytoplasma of the recipient cells. So I will give some details of our strategy. So first we developed the we called VDIC system is co-transfactional two plasmids, VSVG, and one plasmid is EV sorting domain inting cargo. So the EV sorting domain we use CD63 here, and the cargo protein can be any protein of interest. So for the evaluation of the functional delivery, we use the traffic light cells. So for these cells, without Cre-mediated recombination, it will express the IFP, and no GFP will be expressed due to the stop codon upstream. After Cre-mediated recombination, the IFP and the stop codon between the two LOX P sites will be cleaved out and the GFP will be expressed, which can be easily evaluated by fax. So for doing this, we generated several constructs. Cree without anything, no inting, no EV sorting domain, and the CD63 Cree without inting, and the CD63 inting Cree with everything, and the inting Cree without EV sorting domain, and also VSVG. And from the data we can we show here, if we harvest the EVs after transfecting of the different constructs, without VSVG, we did not see any activity in the recipient cells. And only if you have the CD63, the EV sorting domain, and the inting, and also VSVG together, you will get approaching 100% editing in the T47D and the B16F10 cells. This implies that the EV sorting domain and self-cleavage inting, also the phylogenetic protein VSVG, are three key components for the delivery system to work. So then we compared our delivery system with the published nanoblade system. I will have a point here. So the nanoblade is, I think most of you have known, it uses the gag protein from the virus as the scaffold, and it can be fused with any protein of interest, but they have a cleavable peptide in between. So after co-transfection of the gag pore, it will express the protease. So this protease will cleave the protein of interest to be free in the vesicles. So this is how the vesicle like. So after the VLP are formed, the Cre protein or any protein of interest will be free inside of the vesicles. So we compared our delivery system with the nanoblade system, and we got comparable efficiency or even better efficiency in some cell lines than the nanoblade system. So then we were wondering, instead of two plasmids co-transfection, can we simplify the system to only one plasmid? Because like VSBG is also a transmembrane protein, similar to CD63, maybe it will be enriched in the EV surface as well. So we did the single vesicle flow cytometry first. So after transfactor only VSVG ammonia green construct, we observed the majority of the isolated vesicles are VSVG positive. And if we co-transfactor the VSVG ammonia green and the CD63 construct, we can observe similar number of the VSVG positive particles and the CD63 positive particles. This means VSVG is similar to CD63 and it is enriched in the EV surface and it holds potential to function as efficient EV sorting domain. Therefore, we generated this VSVG fold-down inting cargo plasmid. Here we introduced another element, fold-down. It is a trimerization domain. So it will help VSVG to form trimers to function better. So we called this system VFIC, so VSVG fold-down inting cargo system. And we observed the very high efficiency of this system. Even when in 9 EVs, we can achieved around 100% editing efficiency in the B16F10 cells. So previously, the EVs were isolated by TFF. So we were wondering if we further purify the EVs by size exclusion, if the EVs are still functional or not. So after doing this experiment, we can see here, after purification, the isolated VFIC EVs still work well. So that means the potency was preserved after purification. This means different methods can be used to isolate the functional VFIC EVs. So the not most one of the important component of the delivery system is inting. It is supposed to have the C-terminal cleavage activity during EV biogenesis, but we need to show the evidence. So for doing this, we introduced mutant inting variants. One is called H439Q mutation, which will make the pH sensitivity deficient. So this means when the pH drops, the cleavage will not be boosted anymore. And another mutation is N440A mutation. So this is the C-terminal cleavage deficient mutation. So after introduce this mutation, the inting cannot cleave anymore. So it's similar like the direct fusion protein. So we verify the mutants by Western blot first. So after isolating the EVs, you can see if you introduce the pH sensitivity deficient mutation, the cleavage of Cre decreased a lot, and you see the full-length protein. And if you introduce the NA mutation, you can see most of them keep full-length and you have minimal cleavage. And we verify this in the reporter cells as well. So after introduce these two mutations, the editing efficiency in the recipient cells decreased greatly. So from this experiment, we can conclude that our engineered inting performs C-terminal cleavage, and the pH can boost the cleavage. So another component for the delivery system is VSVG, is a phylogenic protein. So to show that the VSVG is crucial for this system, we introduced another two mutants. So one is P127D mutation. So this will make the phylogenic activity deficient for VSVG. And another one is K47D. So VSVG is supposed to bind to the LDLR receptor. If we introduce this mutation, the binding capacity of VSVG will be decreased. So then we generate another plasmid, is CD63 18 ammonia green. So if we only have this CD63 ammonia green, 18 ammonia green without VSVG, you can see after uptake of the EVs, the GFP expression, it's like a dots everywhere. So that means the GFP were entrapped in the vesicles, either endosomals or MVBs. And if we introduce VSVG at the same time, you can see the distribution pattern of the GLP completely changed. So the GLP signal will diffuse across the cytoplasma. That means there are a lot of endosomal escape of the GLP molecules. So if we introduce the P127D mutation here, you can see the distribution of the GLP recovered to the dot distribution again. That means the phylogenic property of VSVG is crucial for endosomal escape. For K47D, K47Q mutation, you can see if we load the same amount of EVs with the same amount of Cre protein, and you can see decreased uptake of the VSVG and the Cre in the recipient cells. That means this FDLR receptor binding is also crucial for the VSVG to go endocytosis pathway. So after delivery Cre successfully, we were wondering if we can deliver more therapeutic related protein or protein complex. Here we tried Cas9 guide ribonuclear protein. So for doing this, we generated the CD63 inting Cas9 and also VSVG fold down inting Cas9 plasmids. And we used the report cells to evaluate the editing efficiency. So these report cells were generated by Oliver Deron's lab, and it works like this. If you don't have the Cas9 guide to introduce indoors, the cells will express M-cherry. If we introduce the Cas9 guide, target the link between M-cherry and the EGFP, it will cause a frame shift mutation. Then remove the stop codon upstream of EGFP, and the EGFP can be expressed. It can be easily evaluated by FACS. So after co-transfection of the VFIC and VDIC plasmid with guide, we can have the engineered EVs loaded with Cas9-IMP. So after incubation with the report cells for different time points, we can observe up to 80% editing efficiency for VFIC EVs. But for VDIC EVs, the efficiency is lower. And we quantify the Cas9 molecules per EV. And we observed around 1.3 molecules per EV for the VFIC system, and only 0.4 molecules per EV for the VDIC system. So this explains why the VFIC system is better for delivery for the delivery of Cas9 complex. So after successful in vitro evaluation of the delivery efficiency, we tried in vivo work in the TD tomato report mice. So these mice normally will not express TD tomato only upon cremated recombination. to remove the stop signals upstream of the TD tomato, the TD tomato will be expressed. So these mice were normally used to evaluate the Cre delivery in vivo. So first we did one ICV injection. So we inject 5 microliter of the EVs into the ventricle, and we harvest the different regions of the brain, cerebellum, cortex, and the hippocampus. And we observed a little bit signal in different regions after we fake and we dig EV injection. And if we do another injection, we combine osmotic pump with ICV injection. We can inject more volume of the engineered EVs. So this pump will administrate around 8 microliter of the EVs per hour, and it will perform 1 day injection. So that means in total 200 microliter of the EVs will be pumped out to the ventricles. So in this way, we can inject much more EVs than the traditional ICV. So after 4 days incubation of the EVs in the brain, we harvest the organs and do single cell isolation, and then run FACS. We can observe up to 40% TT tomato positive cells in the hippocampus and around 30% in the cortex with big EVs. So this efficiency is even comparable to the traditional AAV delivery. So after local injection in the brain, we were wondering if our EVs can work for the systemic injection. So then we did IP injection into the TT tomato report mice, and we harvested the liver and the spleen for analysis, because the liver, spleen are the main organs that will have the main EV distribution after EV injection. Then we observed a significant expression of TT tomato in the liver and the spleen after injection of the VFIC and VDIC EVs. And also we did the co-staining of T cell mark CD3 or B cell mark B220 with TT tomato. And we observed the good overlap of the CD3 with TT tomato, but absent of the overlap of B 220 with TT tomato. This means in vivo, the T cells will uptake the engineered EVs, but not the B cells. So finally, we tried the therapeutic effects of the engineered EVs by deliver the super repressor of F-kappa B. For doing this, we generated some constructs for the VDIC system and also the VFIC system. So the super repressor of the NF-kappa B is the mutant I-kappa B. So it will lose the phosphorylation activity after stimulation by LPS. So it will constitutively bind to the NF-kappa B complex and sequester this complex to translocation to the nucleus. So then we use the report cells we call hack kappa B report cells. So upon stimulation, this kappa B will go to the nucleus and bind to the mini promoter, and then will promote the expression of the luciferase. So after stimulation, you can see clear increase of the firefly luciferase expression. So for evaluating the delivery efficiency of a super repressor, we incubate the report cells with our EVs first to allow the super repressor to bind to the alpha kappa B complex first. Then we added the stimulus like LPS or TNF alpha. And then we observed the decreased expression of the lucifers. This means our alpha kappa B super repressor was successfully delivered into the cytoplasma, and they can sequester the alpha kappa B complex nuclear translocation. Then we perform the in vivo work by using this LPS-induced systemic inflammation mouse model. So we inject the engineered EVs 4 hours before injection of LPS. This will allow the super repressor to bind to the alpha kappa B complex first, and then we inject LPS to induce the inflammation. And after 6 hours of LPS injection, we injected a second dose of the engineered EVs to boost the super repressed delivery efficiency. And we monitor the body weight loss of the mice, also the survival rate. And after 2 days, we sacrifice the mice and harvested the organs. So we can see here the engineered EVs indeed decrease the body weight loss and also increase the mice survival. This means our engineered EVs can have the therapeutic effects in vivo. So because as I mentioned before, the VDIC system is multimodular. So it has 3 components: EV sorting domain, Inting, and the VSVG. And each component can be optimized to increase the efficiency of the functional protein delivery. So for this, we further identified some better EV scaffold than CTCT3. This resulted the PEP2. So as shown here, in this study, we focus on identification of more efficient loading scaffold. So we are not limited to CTT3 for engineering anymore. So the design of this study is like this. We selected in total 55 EV scaffold, which include all the tetraspanines, the 33 human tetraspanines, and the 4 single-pass membrane protein, and 6 late endosome proteins, and 7 cytoplasmic proteins, and the 5 membrane-associated proteins. We used the VDIC system as the Nature Com paper. So after generating all the constructs, did the transfecting and isolate the EVs. We added the different doses of the EVs to the report cells, and we identified the one best EV scaffold is tetraspanin 2. And then we did the functional validation, both in vitro and in vivo, to show that TSN2 was better than CD33 to functional deliver the cargoes. So first we screened all the human tetraspanins, 33 in total in the report cells. We use the same report cells as the Nature Com paper, and the workflow also is the same as before. And after adding different doses of the EVs, we can see 3 of the test spannies are better than CD63 for delivery in report cells. They are TSN2, TSN4, and TSN9. And we validated these results in 3 report cells. And in general, these 3 tetraspanids outperform other tetraspanids for delivery query. And as similar to the previous paper, we also purify the EVs, and we observe the preserve the potency of the EVs after purification. This means again, our engineered EVs can be isolated by different methods, but still they are functional. And then we screen other groups like the cytoplasmic protein, late Anderson protein, memory-associated protein, and single-pass transmembrane proteins. So we can see here in the cytoplasmic protein group, we identified ANXA4 and Syntenine as the best candidates. And for late endosomal proteins, we identified STR3N as the best one. For memory-associated proteins, we can see the myristoylation signal peptide, AIDC1, and the BAMP3. They are the top 3 in this group. And for single-pass transmembrane proteins, ICAM1 and CD316 works equally well in the report cells. So I think this will give some reference to other researchers if they would like to engineer EVs. Uh, apart from the tetraspany, you can choose other proteins as well, depend on the purpose of your research. And then we would like to dig into the mechanism why TSN2 is better than CD63 to deliver functional cargoes. For this, we did the single vesicle flow cytometry as we did before to see if the VSVG colocalization of TSN2 is more than the colocalization with CD63. So for this purpose, we generated the VSVG ammonia green plasmid, TSN2 M cardinal plasmid, also CD63 M cardinal plasmid. And we did the co-transfection of the VSVG with TSN2 and the CD63 respectively. So you can see here, for the double positive population, the like TSN2 and VSBG, they have like more than 23% double positive population for the isolated particles. But for CD63, only 5% double positive populations were identified. That means like TSN2 colocalize much more than CD63 with VSBG. This potentially or partially explain why TSN2 is better than CD63 to deliver Cre or other protein cargoes. Because the functional delivery also will be decided by the cell uptake. So therefore, this cannot fully explain why TSN2 is better than CD63, but they give us a clue to the mechanism. So then we evaluated if the engineered EVs can work well for the primary cells. So we isolate the lung and the bone marrow cells from the TT tomato report mice, and then we incubate them with engineered EVs. You can see we can see here a lot of TT tomato signal will show up after incubation with the engineered EVs for 2 days. And for the HSPC cells, the editing efficiency increased along with the incubation time with the engineered EVs. This means our engineered EVs can work well in the primary cells. Then, at last, we would like to show the therapeutic effects of the engineered EVs. This experiment was similar to the Nature paper. So we delivered a super repressor of the MF-kappa B, and it used the LPS-induced systemic inflammation model. So we can see here the TSN2 engineered EVs increased the survival rate of the LPS-treated mice, and also the EVs can alleviate the tissue damage. You can see the inflammation or hydropic pathology of the liver will be decreased when treating the mice with the engineered TSN2 EVs. So to conclude, our first paper shows that the VDIC and the VFIX system can deliver the protein or protein complex efficiently, both in vitro and in vivo. And they are multi-modular, so a lot of components in the system can be optimized to improve the delivery efficiency. So the second paper, we improve the delivery efficiency by identifying more efficient EV loading proteins. So for future directions, no, because you know the one of the key component is cleavage. So we don't want a lot of pre-cleavage in the producing cells, like because the inting works well at 37 degrees. When you culture the cells, the cargo will be cleaved before they encapsulate into the engineered EVs. So now we are working on using the mutant inting to improve the cleavage. So we focus on the Cas9 RMP delivery. So another point is EV scaffold. Even though we identified TSN2 as the best scaffold for now, we identified even better scaffold for base-edited delivery. And for VSVG, maybe instead of VSVG, we can identify some more efficient human-derived fusogenic protein to avoid the potential immunogenicity by using VSVG. We tried to screen 40 human-derived fusogenic proteins in the Nature paper, but none of them worked. So maybe other researchers can dig into this. If they can find or they made some modification to the human-derived fusogenic proteins, they can work equally well as VSVG. This will be much beneficial to the system. And the last point is like our work may inspire other research to develop even more advanced delivery system. Because you know, one of the aims of the publications or doing research is to inspire others. So they can improve our system or they can develop a complete new system in future. So at last, I would thank to my colleagues in Andalusis group and Evox Therapeutics to support the studies and other collaborators. Thank you for your listening, and I'm happy to take your questions.
Well, that was a real tour de force there. Thank you so much, Xiuming, for presenting this. I don't think there's any question that you're inspiring people here. That's really great. I want to just start just by reminding everybody, please put your comments and questions in the chat box, and then I'll call to you in the order that they're there. I do want to start with a question about how you think we should go about identifying better scaffold proteins. Is it going to be just a matter of screening the proteins that we already know, or does there have to be also some engineering component that might go even as far as creating novel synthetic proteins?
I think that's a good point because, we can take the test spanning CD63 as an example. So the Y type CD63 maybe is not as good as TSN2 in our study, but if we have the mutant CD63, like other people did, they have this Y235A mutation, it will increase the loading of the cargoes. Maybe if we introduce such kind of mutation to other types of spannings or other groups of proteins, it will increase the loading. So there are a lot of work to do to identify this. So our group is one of them to do this kind of work.
Great. All right. Well, I see that we have a lot of questions that are coming in here in the chat box. So we're going to start with Phil Askanase. Please go ahead. I will read Phil's question here. So delivery of more than 1 to 2 copies per cell, delivery to non-lymphoid cells in vivo, and delivery beyond 1 to 2 passes. So how do we increase the delivery, make sure that we're delivering more molecules?
Yeah, that's also a challenge point. So we should date that for CRISPR-Cas9, now we only have like maximum 1.5 molecules per EV. So they have a big space for improvement. So for VLPs, they published around, if I remember, 100 molecules per particle, right? So if we can reach that level, I think we can increase the efficiency by 100-fold. So for EV field, I think still there are a lot of space for improvement.
And then the question about delivery. So you inject into the bloodstream, you might have those EVs sticking around for 1 or 2 passes. Is there some way to improve that? I know that your group has done some work on that in the past as
Yes,
well.
I had done that before. We used the albumin binding domain on the EV surface, so they will bind to albumin after injection, and this will extend the circulation time by more than tenfold. So we published that in GF in 2022. So we can combine that strategy with our currently engineered EVs. So to achieve the circulation time extension or also functional delivery at the same time.
That's the beauty of EVs, right? They're a
Yeah,
scaffold,
yeah,
so
yes,
you're not limited to one strategy or one challenge with a single EV. You can address a lot of them at once. So
yeah, yeah.
thanks, Phil, for the questions there. Hujan Kai, please go ahead.
Uh, okay, so I have a question. So since you mentioned that it's a 37 degree in the EV producing cell, so
Yeah.
did you measure before in the EV producing cell how much is being cleaved before being packaged into the EV? You know, the percentage of this fused protein, how much? So what I worry is if you put the VSVG into the EV producing cell, they produce a lot of these particles, so they will non-specific pick up your packaging, the Cas9 will create. You know what I'm saying? Um,
Yeah. Yes, I think we had one Western blotted data in the producing cells. So we did not quantify, but it means the pre-cleavage, right? The
yeah.
like cleavage before encapsulating into the engineered EVs. There are quite a lot, I can say, because like they can work very well at 37 degree. So after transfection of the plasmid, uh, it will be translated into a protein. So then it will start the cleavage. So and after the pH drops, it will boost the cleavage. So that still means there are a lot of cleavage before encapsulation into the EVs. So our current work, one of them is to mutant this intin. So it will decrease a lot the pre-cleavage. So we now have a new intin that can minimize the pre-cleavage like to not to near to zero. So all of them will go to the vesicles.
So are you able to measure the pH in the inside the cell? This is I cannot imagine how you're going to how to
No,
measure the pH.
no, the
But you
answer is no.
assume that is in this pH low pH that environment. This is based on
No.
assumption, right?
No, the answer is no, we cannot.
Yeah.
But the mutation is referred from one publication. They did in vitro work. They have the antin and they have this like H439Q mutant, and they observed the decreased cleavage, even the pH drops. So from that paper, I introduced the mutation in my study, and we observed the same, both in the producing cells and also the isolated EVs. Also, if you incubate the engineered EVs with report cells, you observe huge decrease in the editing efficiency. So that's not directly tell us it is real pH dependent, but we guess like in the most property, in the most case, it is pH dependent, especially like during the EV biogenesis, they have the late endosomal environment with the pH like 5.5 to 6, so it will boost the cleavage in that environment.
Good job. Good job. Congratulations.
Thank
Thank you.
you.
Thank you, Hujan. And I want to apologize to everybody for the technical issues here. I'm not sure why the controls are not working for unmuting. If you are able to unmute, please feel free to jump in. But in the interest of time, I think I'm just going to go through these questions here. So, Xiuming, Matthias Ostrowski asks, do you have any clue why the HERV fusogenic proteins didn't work so well? Are they full proteins? Because a lot of these HERVs, they're ancient ancient
Yeah,
insertions into the human genome, and some of them have been mutated or aren't full length anymore.
I think they even though they are endogenous, they are derived from the virus long time ago, but they are humanized, so they are educated by the human environment, so they cannot have a lot of phylogenic property anymore. That will cause dangers to the human body, I think. So they are changed to some extent to like impair the phylogenic property or change something. So we don't know. So maybe one direction is like if we can recover its original shape or original configuration, like a 3D structure, and they have the strong phylogenic property. Maybe one of the like endogenous progenic protein will work, but this will demand a lot of work, not only from our groups, other research can also dig into that.
Yeah, it's a lot of opportunities there, to try to
Yeah.
reconstruct those ancient proteins and their properties. Then, of course, you have the potential issue of immunogenicity again,
Yes.
because now it's different from what it was. But if you can
Yes.
somehow get around that, so lots of interesting possibilities here. And Zach Troyer also notes in the chat box, that in his experience, there's sometimes expression issues too with these proteins, and that can sometimes be solved by flanking with certain sequences. So there's a lot of work that could potentially be done here. So that's great. We do have a question about PhD positions, so I would urge anybody who's interested in the Andalusi lab, please feel free to reach out to Samir or Xiuming about that. Next question here is from Guido, who says the GFP reporter response is likely not EV dose dependent since the reporter is either on or off. Therefore, what is it magnitude and process is really measured? And what about non-gene editing cargo? So how can you look at the magnitude when you have a binary response?
So I did not totally get this question. So is not those response, or what's the main point?
So as I understand it, the lack of dose response might simply be due to the fact that you have a binary, it's either it's on or it's off. So what are some ways that you could look for magnitude of response? Maybe not using that reporter, but are there other reporters that you could use?
But I think regarding this, maybe we can use we can use endogenous target instead of the report cells. Maybe for CRISPR-Cas9, we can target BCL11A or some interesting target, not like report cells. Because for the report cells, it's easy. It's a tool. It's easy to evaluate, but it was generated by lentiviral transduction. So maybe that will have, you don't know how many copies per cell, right? So it's hard to say. So, but for endogenous target, you only have two at least. So that's easy to be interpret.
Very good. A question here from Cecilia, who thanks you for the presentation and asks, what about strategies to maximize target cell specific delivery of engineered EVs? I know that you showed that for some of your constructs, you had T cell uptake, but not B cell uptake. How do we go about that?
I think for our engineered EVs, like we have the decoration of the VSVG on the surface, right? So the tropism of VSVG will decide the cell specific. So maybe for T cells, they have more LDLR receptor expressed, but not the B cells. So that's why the T cells uptake more. But for future study, if you really want to do the target delivery, you can degrade the nanobody or some target moiety on the EV surface, and you can use the mutant VSVG. So that's like already published by other groups. So this is a strategy. You can achieve the like targeted delivery. You can target whatever cell types you want, right? You just change the nanobody or change the targeting moiety on the surface.
Great. Okay. And we'll go with one last question here, which is it possible to utilize EVs from biological origin? So I'm assuming, you take somebody's plasma EVs or EVs from some other source and encapsulate drugs or chemical compounds similar to how liposomes are used. So this is beyond what you have, or a completely different, I think, approach, the post-production approach.
Yes, this is different from what we have done because we are focusing on endogenous engineering. So we normally engineer parental cells and to produce the engineered EVs. But this is the post isolation. Like after you harvest the EVs, you put something functional cargoes into the EVs or inside or on the surface. So I think it's possible, but right now it's a technical challenge because for loading, let's say siRNA, people use electroporation or some sonication or something, but it's not efficient, I can say, or it will cause some problem like the EV aggregation or some other issues. So right now it's feasible, but I think there are a lot of challenges.
Yeah, and I would just note that there has been some literature on this, including from some of your co-authors, like Matthew Wood's group did the work with GAPDH. You can have things that are binding to various elements on the surface of the EVs, including phosphatidylserine, for example. And you can load things at least on the surface that way. So
That's true.
again, the more we know about the biology of EVs, the more we can exploit that to make the EVs do what we want them to do. So thank you again, Xiuming. Thank you all for also staying a couple minutes over here. This has been a very educational presentation, and also an inspiring presentation. So it
Thank you
shows
so
us not
much.
only all the great work that you've done, but also all the many things that remain to be learned about EV engineering. So let's all get back in the lab and do some more great work. So thanks again, Xiuming. Thanks everybody for
Thank
joining
you so
today.
much.
And
Thank
we
you.
look forward to seeing you again on a future EV Club.